And then we can live here. Hello, everyone. I'll let people get in a little bit, and then we're going to start in about a minute or so. All right, so I want to say hello to all of you for our second day of Global Coral Reef Week. My name is Franziska Elmer, and I'm an independent researcher who is part of the organization committee. And together with Chelsea, my co-organizer, who works in Hawaii as a postdoc, we will do the technical support today. And Gustavo will lead through the program. Feel free to use the chat today to introduce yourself to the other people. So all the attendants have access to chat and you can talk to each other during the meeting. So don't be shy, talk to each other, get to know each other. Please use the Q&A chat at the bottom for any questions you have. We will have time for questions at the end, and some of your questions can be asked. You can also upvote questions. So if somebody asks a question you really want to hear the answer to, you can vote it up, and therefore it will be on top of the list to be asked. If you are having any technical difficulties and nobody else in the chat is writing about them, then it's probably your computer or your device. And we suggest to you that you try to log out and log in again. If that doesn't work, we also are live streaming this on YouTube, so you can watch it on YouTube or rewatch it afterwards. To start, we want to get to know where you're from, and do a little poll of where you are located right now. So you can see a poll now, and you can fill out where you are from. Seems like either nobody is filling it out or we're having some technical difficulties with it.
Fran, I'm seeing we have about 86 people voted, and it looks like 39% today are joining us from North America, 26% from South America, and then we've got a good handful of people from Europe and the Caribbean and the Mesoamerica coming in.
Okay, I couldn't see those polls, but you could see it, so that's perfect.
Yeah, I can click end poll if you think.
Yes, I think we're good.
Okay.
And then we have a second poll for you. Here we see the results as well. Yes, most people from North America and South America, but quite a lot from Europe and Caribbean as well. Our second poll is about where are you working? So which sector are you working in? So we kind of know what kind of audience we have. giving you guys a bit more time. And let's see the results. A lot of people from academia, lots of students, about 30%. But as yesterday, conservation management and government are also well represented. And also a few people from tourism, education, industry, and very exciting. All right, so without further ado, I want to give the microphone to Gustavo, who will tell you a bit about today's program.
Hi, everyone. I will introduce the professors here. So we are going to start with Dr. Godoy, who is an associate professor at Federal University of Rio Grande do Sul, Brazil. His research is focused on biotechnology applied to reproduction of aquatic organisms, development of cryopreservation protocols, and the effect of low temperature on metabolism of reproductive cells. He is an associate researcher at Coral Reef Research Network and leader of the Reef Bank project, which aims to investigate the physiology and biochemistry of coral gametes and use of cryopreservation techniques to create a frozen bank of gametes from Brazilian reef building corals. Last year, Dr. Godoy received the Young Investigator Award from Society of Cryobiology. He will be followed by Dr. Costa Lotufo. He's a full professor and head of Department of Pharmacology, Institute of Biomedical Science, University of São Paulo. Her lab is focused on the discovery of new anti-cancer compounds from Brazilian marine biodiversity and studies on their mechanism of actions. Recently, she started a multidisciplinary project to access marine microorganisms diversity and biotechnological potential along Brazilian coast and islands. Dr. Costa Latufo's activities include the supervision of undergraduate and graduate students and postdoctoral fellows. She has published more than 270 articles in peer-reviewed journals. On 2017, Dr. Costa Lotufo was nominated as member of the Academy of Science of São Paulo State. Following the speakers, Luis Rocha is an associate curator of the Follett Chair of Ichthyology and the California Academy of Science. He is also an adjunct professor at the University of California, Santa Cruz, and San Francisco State University. Rocha has authored one book and more than 100 scientific articles. He is best known for his work in speciation of coral reef fishes using advanced genomic methodologies, and recently has been actively exploring the diversity of deep coral reef through the topics. And the last speaker will be Raquel Peixoto. Is associate professor at the Federal University of Rio de Janeiro, Brazil, visiting professor at UC Davis, and research associate at Rio de Janeiro Marine Aquarium. Her group's research has outlined the protocols and proved the concept that the manipulation of coral-associated microorganisms, known as beneficial microorganisms for corals, is possible and can increase marine host resilience and resistance against environmental threats. This pioneering work has contributed to pave the way for the new approach to reveal and explore mechanisms of marine symbiotic interaction, including other marine organisms. We will start with Godoy's talk, and at the end of the four talks, we will start the questions and answers. Dr. Godoy, can you start your presentation?
Thank you, Gustavo. Can you hear me?
Yeah.
I'm going to share my screen. Can you see my presentation? Is it okay?
Yes, it's okay to me.
Okay. Thank you very much. I'd like to highlight the amazing work of the Brazilian committee for this meeting. It's a pleasure to talk about the Brazilian coral reefs and to show you some of the reproductive biotechnologies we are studying at Coral Reef Research Network. There's something very wrong with the planet. We are going through a difficult time, but coral reefs have been facing their own pandemic for several years. An entire ecosystem is collapsing as bleaching episodes have become more frequent and more intense. Until 2018, over 300 bleaching episodes were recorded in the Indo-Pacific and Caribbean reefs, with mortality being reported virtually in half of them. As a result, several initiatives have been tested worldwide to help saving coral reefs. For example, coral gardening, increasing marine protected areas. However, if we keep burning fossil fuels and interfering with the planet's climates like nowadays, none of these initiatives will help. Some daring initiatives like assisted evolution are under investigation as well. And my goal today is to present you some additional approach that can make a difference in the fight against extinction. Joint forces and expertise could indeed make a difference. But before any biotech, it's important to say the time has come for the world to realize the importance of Brazilian coral reefs, as very well described in this just published paper of Dr. Mies and collaborators. Unlike others, South Atlantic corals evolved in a turbid and nutrient-rich waters, and these local conditions resulted in such a high trophic plasticity, which has been crucial during bleaching. Even during the most intense bleaching event recorded last year in Brazil, with bleaching achieving 90% of stony corals, mortality rate did not exceed 2%. We witnessed an unprecedented episode in which colonies of Mucismilia hart and Mucismilia ispida spawned while fully bleached. So I'm talking about these resilient broadcast spawners in which sexual reproduction play an important role, promoting genetic diversity, as those corals who survive extreme conditions can generate offspring as strong as their parents can, being crucial for larval input and recruitment to guarantee reef survival and recovery. But what do we know about coral gametes biology, biochemistry, chemotaxy, ultrastructure? We should pay much more attention to these gametes. I mean, not only taking a quick look at the oocytes and spermatozoa, but to investigate the physiology and biochemistry behind them. Sperm motility rate means the percentage of live motile cells. In this case, of Mucismilia ispida, spermatozoa can survive up to 22 hours after spawning. For Mucismilia hart, the spermatozoa lifespan can reach, as you can see, up to 16 hours after spawning. If we look at the spermatozoon by light microscopy, we will see a common head shape and long flagellum. However, when we use transmission electron microscopy, look how fascinating it is. Here we can see the head of the spermatozoa, the central nucleus, the Golgi complexes, and look at these two huge mitochondria which generate the energy needed for the flagellum bead. In the case of hermaphrodite species, the gametes are released in the water in this oocyte sperm bundle. The distribution of spermatozoa and oocytes is not uniform in the bundle, with these dark purple areas representing regions with sperm. But perhaps the most interesting thing we discovered is that this bundle is not just a ball of mucus wrapping the gametes as was thought. In fact, the bundle has a structural organization. In fact, look at these hexagonal shaped structures similar to beehive alveoli in which sperm are contained. Each of those are spermatozoa enclosed in this alveoli. Additionally, this structure is able to keep the sperm immotile while not coming in direct contact with the ocean water. What about the attraction and biochemical conversation between the oocyte and the spermatozoa, which ultimately leads to fertilization? Believe me, this has been investigated in just 4 species of stone corals and will be one of the focus areas of our research in the coming years. There are several biological markers such as cellular, structural, and biochemical components which can define the status of an individual or even be a response to high-intensity stressors such as bleaching. Here, you can see a simple assessment of sperm lifespan in Mucismilia heart in 2018, a year without bleaching episode, compared to 2019, a year of intense bleaching. And even though the colonies have recovered from bleaching in October 2019, the sperm's lifespan has dropped to half, as you can see. But how water temperature affects coral gametes? In this experiment, we exposed the spermatozoa of Mucismilia heart to 3 different temperatures: 26, which was the temperature at Recife de Fora Marine Park, an increase in 1.7 and 4.8 degrees, according to IPCC projections of future ocean conditions. And clearly, we see that higher temperatures, spermatozoa were already dead after 5 hours. What's going on at biochemical level? Reactive oxygen species are molecules produced in normal metabolic processes, such as cellular respiration. However, when there is an imbalance between the generation of ROS and antioxidant capacity to neutralize these compounds, a state called oxidative stress is created. We know, for example, the symbiosis breakdown is a result of oxidative stress, but how oxidative stress affects coral spermatozoa? This drop in reduced glutathione concentration indicates a condition of oxidative stress and finally cell death, which can be confirmed here when we look at T-bars till barbituric acid reactive substances concentration, a lipid peroxidation biomarker also indicating oxidative stress, which is higher at higher temperatures. But finally, I would like to introduce you to the cryobiology world. Cryopreservation has been considered one of the most effective tools for species conservation, since at cryogenic temperatures, all cellular metabolism is completely paralyzed, and this biological material can remain stored for decades, safe from any catastrophe. However, developing a cryopreservation protocol is not an easy task. It requires knowing the sensitivity of that particular cell and the most appropriate cryoprotective agents to guarantee cell survival. We have been conducting several experiments aiming to create a bank of coral gametes. At this point, we have hundreds of samples frozen in the laboratory, waiting for the pandemic to pass before resuming analysis. Once an efficient cryopreservation protocol has been reached, we need to perform in vitro fertilization to generate new coral babies. So graduate students are going to conduct these experiments in the coming years. To conclude, my final message is, let's explore biotechnology and joint expertise for coral reef conservation. I'd like to acknowledge the vital partnership with Coral Vivo Project and Coral Vivo Institute. Without them, none of this would be possible. A special thanks for Boticário Group Foundation and the Brazilian Fund for Biodiversity for their financial support. And thank you very much.
Thank you, Leandro, for your talk. Very nice talk. And to follow the presentation, I will pass through to Dr. Costa Lotufo to start her presentation. The questions will be in the end. Sorry, your audio is off.
Here we go. Now you can listen to me?
Yeah,
okay.
Yeah, okay. Thank you.
Okay. Thank you, Gustavo. Sorry about that. First of all, I want to thank you for the invitation. It's an honor to me to be here discussing with this audience some of the research that we do in our lab. And also, I had the opportunity to follow the event yesterday, and I may say that the presentations are actually quite interesting. So after this very beautiful presentation from Leandro, showing us a possibility, how can you take care and preserve and cryopreserve the corals, I'm happy to see how forward we are in this technology. I will migrate for a completely different subject and trying to present you how can you search for marine natural products in corals using sustainable approaches. And if you think diversity, we will see that we are more familiar to study diversity as this beautiful, like colonies and colorful and bright species that you have in the ocean. But I want to invite you to look a little bit more for the whole molecules that this diversity can produce. And in fact, I hope to show you that we can have a look in all this, what we call chemical diversity, as myself in the pharmacology lab, looking at these molecules as possible drugs prototypes, but we should look this as also how all these species communicate. So we can have a look in the chemical diversity from a coral perspective. And all this, the host, the invertebrate, the whole microbiota that I believe that is what Raquel will talk to us in the last presentation. This association has very important implications in the coral health. This is not only true for corals, but it's true for humans and it's true for everybody, that we are actually intricate ecosystem. And this talk about the symbionts and the hosts are important if we think about nutrient cycling, the chemical defense, the resilience. This is being incorporated, I think, in the conservation studies, and it's bringing some very important points. How can you look conservation? But if we look this from a human health perspective, we may see that most of the medicines that we use, like 60%, or if you think cancer, more than 70% of the medicines actually come from a natural product. It's inspired in a natural product. In these slides, you can see that even the statins, the medicines that we use to treat heart disease and low cholesterol levels, blood levels, come from fungus. And what I want to show you is that if we think about the marine environment, we do have some examples. Now we have 13 medicines that you can find in the pharmacy. Most of them, you can see here 4 in the slides, that are used to treat cancer. And in the past 2 years, we have 4 more that were approved by the health agents. And this has a great impact on the treatment of this very high mortality associated disease. In fact, when we think about all the diversity that we expect from the ocean, we can anticipate that we can develop, we can find almost 600,000 new compounds related to this diversity. And this can bring us more than 200 new anti-cancer drugs. But what I think is crucial to bring to this picture is not values, but how can you learn from all this evolutionary story that we have, that went through in the oceans, and bring innovation without destroying the environment. So we have many ways to assess the chemical diversity. We have a more like classical approach that you only search whatever the coral produce. We have more like a disease-oriented approach. When you have a disease that you want to try, and you can use some cell-based assays or phenotype-based assays to find these compounds, or you can use proteins to fish compounds from these environments, invertebrates, and find out new drugs. And you have a more like intricate or complex approach that is what we call the omics approach, that we can try to understand patterns, complex patterns that emerge for the whole holobiont, and to understand how this bacteria, metabolites, and hosts, they talk to each other, how this has an impact on the species fitness in the environment, and how can you looking for this whole like complex data, how can you find out also new prototypes to develop drugs? Our lab has we've been studying this for the past probably 15 to 20 years, and we are using all these approaches to find new anti-cancer prototypes. We have a very nice partnership with the Sysbiotomar network and PELG, Oceanic Islands, the program for ecological monitor Kadu talked to us yesterday in this meeting, and I think Sergio Flutter, that is the coordinator of Sysbiota, will talk to us tomorrow. Just to bring you some examples of what we do in the lab, we've been working with CMAT corals from Palitoa species. That is the most common ones in Brazil. Duda presented us this diversity yesterday, and they are known because they produce a huge toxins that we call palytoxin. And since the 70s, people are trying to assess the chemical diversity of these animals. We can see that already many molecules have been isolated from these animals. And some of these molecules, as we can see here, like aliphatic amino acid, steroid, they presented anti-cancer potential. But this is using the conventional approach where you take the invertebrate, you take an organic extract, and you follow through the activity. This is not sustainable. We need more sustainability in this research. So we are trying to use the Palitoa using like not the coral itself, but the microbiota that we can take from this coral that we believe not only add the sustainability, but also multiply possibilities to find new anti-cancer leads. So what we do is we take the corals, we isolated the bacteria that we can cultivate from these organisms, and using like common microbiology techniques, we isolated more than 60 strains from the Palitoas, and especially this one that is a Streptomyces strain. We could isolate this class of compound. I'm not going deep on these results, but just to show you that from the coral, we just take a tiny small amount, like 5 milligrams, and we isolated sometimes over 100 bacteria. And for each one of these bacteria, we can take many molecules. And in this case, we showed using a very important protein in melanoma, the skin cancer, we find out that these molecules can actually target an important transcription factor, and this gives us an anti-proliferative potential to treat cancer and anti-metastatic potential. Just to show you that how far we can go in the cancer research. And using the omics approach, what we've been showing, we're trying to put together the toxicity to cancer cell lines, the metabolomic studies, and the metagenomic studies. And what we have so far using these two species is we use a whole bunch of metabolomics techniques where you're trying to show everything that these corals produce. Of course, we never get everything, but we have a very complex data. But for our surprise, when we try to analyze and group this data, the two species are more related when they come from the same environment. Then the same species had been collected in different places. So apparent metabolomic is not as simple as we thought to be using chemotaxonomy, because we have a high influence on the environment, on these molecules productions. And we did show that the production of these metabolites related to some environmental conditions. But the taxonomy of the microbiota, if you see here, it's perfectly related. No matter where I collect the samples, I have a very conservative microbiota. And when I disrupt this relation, actually, we have observed that this is it's important to many diseases that we can observe in the corals. Just to show you that this is go a little bit further, showing also the functions of the genes that we can find in this microbiota community. And finally, to finish this talk, I just to show you that the corals, and I think Raquel will talk about this, they have a very intricate relation to the microorganisms, but the production, the chemistry resulting from this talk is highly affected by the environment. So when we change the environment, when we have like the global impacts, we will also change how the marine species are talking to each other. So just to finish, we have a bunch of collaborators, a very happy team in the lab, and I want to acknowledge all the funding that my group received. Thank you.
Thank you, Leticia, for a nice talk. We will follow to the next talker. It will be Luis Rocha, that will talk about the new techniques, new technology in dive studies. Luis.
Hi, everybody. Let me share my
Luis.
screen here.
Thank you.
Okay, can you see it okay?
Yeah, it's okay. Thank you.
Okay, hopefully there's a video in there in the middle. Hopefully when we get there, the audio will work. We tested it, so it worked before. So this is gonna be very different from the other two presentations, and probably the last two. I'm not gonna talk anything about biochemistry or enzymes or microbiology. My talk is more related to a different kind of technique, more of a diving technique that we use to explore those deeper reefs throughout the tropics. And before I go too far into it, I wanted to explain what do I mean about deep reefs. So the technical term we use for this particular region is mesophotic. So what we're talking about here is mesophotic coral ecosystems. There's many different ways to define them. The official depth range is from 30 to about 150 meters. But depending on how clear and how warm the water is, they can start a little deeper. So maybe 40, 50, 60 meters. So that top portion of the reef, the top 30 meters or so, is the portion that's highly accessible, that every scientist studies. You only need like a open water scuba training to dive to about 30 meters depth. It's very well studied. So we know a lot about that top 30 meters of the reef because a lot of scientists have access to it. If you want to go deeper, there's a few options and none of them is very straightforward. Because of that, there's these deeper portions of coral reefs. They have been completely understudied, and they are, in most parts of the world, they're almost completely unknown, with very few exceptions like Hawaii, which is a place we know quite a lot about the mesophotic ecosystems, because there's a lot of people using these techniques and doing this type of diving there. But outside of Hawaii and maybe the Northern Red Sea, we know very little about those depths. So the depths I'm talking about are between 30 and 150 meters depth. And they're very hard to explore because they require you to do it using technical diving. And for technical diving, you need a lot more training than the normal just open water diving. For open water diving, you just do 1 week of training and you're done. For technical diving, you need to do several levels of training, and you need to accumulate a big number of hours diving in order to show that you can do that by yourself, basically. That keeps a lot of scientists out of this just because it's so hard to get to. Now, a few people try to study this using submarines or ROVs, and they're okay to study things like corals or gorgonians or other animals that don't move. But my specialty is fish. And to study fish using a submarine is very, very hard. It's like trying to study birds on a rainforest using a helicopter. So when you get close to the mesophotic reefs with the submarine, with all the lights and the noise and the vibrations that the submarine does, all of the fish hide and you can't really see them. It's the same with an ROV. So the best way to study fish at those depths is really by diving. And that's what we do. And there's a few different ways you can do it. The first way is just the more gear. So when you're doing a normal open circuit dive, you just have a tank and a regulator. To go deeper, you need a lot more gas. So that's what those pictures show there. They show divers with a lot more gas. You need mixed gas. The deeper you go, the more your body absorbs the gases, and those gases, they interact physiologically with your body in very different ways. So you need different types of gases. We breathe a high percentage of helium at depth. So when we go to depths like 150 meters, we need to be breathing a gas mix that is upwards of 70–75% helium. If we breathe nitrogen, or if we breathe just compressed air at those depths, we get, oxygen becomes toxic, and nitrogen becomes narcotic. So it's a big no-no. You have to breathe different gases, and the different gases have to be in different tanks. And that brings up one of the biggest disadvantages of using the normal scuba gear to do this, which is if you have different mixes of gas at different tanks, those mixes, if you breathe them at the wrong depth, so say you have a tank that has a mix of 75% helium that you can only breathe at the very deepest portion of your dive. If you breathe that at the surface, you pass out and you drown. If you breathe your shallow gas that is high in oxygen at depth, you pass out and you drown. So if you do, so you see all of those tanks there, they have different regulators attached to them. If you pick the wrong regulator and put it in your mouth and breathe for 2 seconds at the wrong depth, it's fatal. It can be fatal, and accidents happen. So the other disadvantage of using this type of diving, which is like open circuit diving at depth, you can see it in the photo on the right there. So when you do normal scuba diving, everybody that saw a documentary that dove themselves, you know that when you exhale, all of the gas comes out as bubbles. It goes back in the atmosphere. So you inhale, you breathe in the gas from the tank, and when you breathe out, when you exhale, all of that gas goes out as bubbles. The deeper you go, the more gas that is, because the deeper you go, the denser the gas gets. So the more gas you need to fuel your lungs. So at depth, at the deepest portion of the dive, at about 150 meters or so, you can empty one of those normal cylinders, normal scuba tanks, in about 3 to 5 minutes, depending on how hard you are working. So that's how much gas you are using. And helium, so as I just said, at those depths, you need to breathe a mix that's 75% helium. So helium is a very expensive gas. You put a lot of it to fill that tank, so you're spending a lot of money. So the first disadvantage of this type of diving is that it's not really safe. Any mistake can be fatal. The second one is because you spend a fortune in helium. That really adds up the more dives you do. It can cost as much as $5 per breath at really the deepest portions of the dive. So the solution we found was that one on the diver on the right there, that's Yanni, one of our dive buddies, is using a rebreather. And the rebreather technology offers us many different advantages to dive at those depths. Before I go into them into detail, I'm going to show you how it actually works. So that scheme there represents a diver. As I just said, when you're diving with the open circuit, with the normal tank, when you exhale, all of the gas comes out with the bubbles. In the rebreather, it's different. When you exhale, all of the gas goes back into the system. Now, the problem is when you exhale, so when you inhale, you take the normal air, right? And then you use the oxygen in the air to metabolism, and that becomes CO2, and you exhale CO2. If you inhale that CO2 back, that's bad for you. So what happens? So when you exhale, all of that gas that you exhale goes back into the system. There's a filter in the back of the rebreather that removes the carbon dioxide, removes the CO2, that's byproduct of respiration. Then there's 3 oxygen sensors. So there's 2 problems there with the breath that you exhale. One is that there's a lot of CO2 in it. There's a lot of carbon dioxide in it. That's the byproduct of respiration. And two is that because you used oxygen, when you exhale, there's less oxygen than when you inhale. So we solved that. There's 3 oxygen sensors on the back of the rebreather that constantly monitor the pressure of oxygen in the gas that you're breathing, and it only adds the amount of oxygen that you need. Right? So it removes the CO2, adds the oxygen that you need, and then it gives back to you purified air, basically, with a breathable amount of oxygen and no carbon dioxide, because it filters all of the carbon dioxide out. It's essentially the same system that astronauts use in space, because they don't want to be carrying those giant scuba tanks to breathe air. So they do the exact same thing. It's a very efficient system. And with 2 small tanks on the back there, you can spend, in the rebreather that we use, you can spend as much as 7-8 hours in the water. The limit is the amount of oxygen in the oxygen tank and the duration of the filter, which is usually 7 to 8 hours. So let me go back to the other photo there. That's Yanni again. So those tanks that you're seeing under his arms there, we never use those. Knock on wood here. We never use those. Those are just bailout tanks in case the rebreather fails, because all of those oxygen sensors, they're electronic. So if they fail, then we need to switch to a system where we know how much oxygen we're breathing, which is in this case, the bailout tanks, the emergency tanks. But we never, I've been doing this for 5 years. We've never needed to switch to bailout except for very few circumstances. So they're just for emergency. So again, inhale, take CO2 out, add oxygen, breathe again, same gas. So what happens here is that you see that in this whole diagram, there's no helium, right? Because nothing is happening with the helium. The same helium you're breathing over and over again. So 2 advantages. One, we don't need to switch the regulator and go to another tank, potentially getting a tank by mistake and breathing the wrong gas at the wrong depth. And 2, because we're breathing the same helium over and over again, this is way, way cheaper than doing an open circuit dive using a normal scuba tank to go to those depths. Now, the unit that we use is this one, is the Hollis Prism 2. There's 5 or 6 or 7 manufacturers out there. I like some better than others. This one we use simply because it was the company that was based here very close to us in Oakland, just across the bay from San Francisco. And that's the one we started using 5 or 6 years ago, and I really like it. It's very sturdy. We travel all over the world with it. Never had any problems with it breaking during airport transports and everything. And I really like the electronics. So on the right side there is the diving computer that tells us all the information we need to know during the dive. So those 3 numbers there that you're seeing in the middle of the computer, they're the most important numbers for the whole dive when you're we are diving with a rebreather. It's the partial pressure of oxygen. So right now it says, I can't really see the one in the right there. Yeah, now I can. 1.23, 1.25, and 1.36, right? So those are the 3, there's 3 oxygen sensors in the back of the rebreather. Those are the readings I'm getting from the 3 oxygen sensors. They don't completely agree because they're different sensors, so they read the oxygen in a little bit different way. In this case, the 1.36 there is significantly higher than the others, but not so much. So we still can make an average there and say that we're breathing more or less 1.3 oxygen or so, something like that. If that number, if the oxygen goes too high, or if it goes too low, it can be fatal to the diver. So because it's so important that we know those numbers constantly, the most important thing we do during the whole dive, more than collecting fish, more than counting fish, more than collecting corals, more than all the science, is looking at those 3 numbers there through the whole dive. We have to know exactly how much oxygen we're breathing the whole entire dive. So back, I don't know, maybe 2-3 years ago, the way we did that was every 30 seconds or every 15 seconds, we look at that, we bring our arm up and look at the partial pressure of the oxygen. Otherwise, it can go up very quickly and it can go down very quickly, and it can cause really big problems during the dive. So one of the super exciting technological advances in the whole rebreather diving world was the introduction of this, this computer here, this thing here on the right. So that's, I don't usually do selfies underwater. That's me in St. Paul's Rocks, but I did that because my hoses, the rebreather hoses, were pulling, and it was I had to hold my rebreather in my mouth really strongly, and I didn't know what was pulling the hose out. And my jaw, those dives are really long. They can be upwards of 5, 6, 7 hours long because of decompression. So my jaws were fatigued, and I had no idea what was pulling the regulator away from my mouth. So I took a selfie like that to look at it after the dive, and then try to fix and see which hose was pulling what. And I realized that was the hose that was wrapped in that yellow band there. But what I wanted to look at is this thing on under the red circle there. It's a it's a Shearwater NERD computer. So that computer, it's right in front of my eye, and I can look at it without even, I can have it on my peripheral vision without even paying attention to it, and then immediately knowing everything that's going on in the dive. So that one on the right is the one that's on my wrist, and then one on the left there is the Shearwater NERD computer, the one that is right on my eye. So all of that information that I need to do during the entire dive is right in front of my face. I don't even need to look at my wrist anymore. So this computer allowed me to do, I don't know, 5 times more science than I did before in those deeper dives. Because when I was doing a transect or taking pictures and having to look at my computer every 10, 15, 30 seconds, it was really taking out a lot of time, especially because when you go to 120, 130, 150 meters depth, the most you have of science time, of working time at those depths is 3, 5, 6 minutes. So if you have 6 minutes and you have to lose one of those 6 minutes just looking at your computer, that's a whole 20% of science you could be doing in that dive that's gone. So having that computer right in front of my eye means solve a million different problems for me, in addition to making the whole operation 10 times safer, because now I can look at it every 3 seconds instead of every 15 or every 30 seconds, and without stopping everything else that I'm doing. So that really, really helped us. And it allowed us to do a lot of different things like Hudson is doing. Hudson is was my PhD student, now a postdoc with us here at the Academy. Well, he's physically in Brazil now, but we're still collaborating a lot in several different papers and work that he's done during his PhD. He's doing a transect there. At the time I took this picture, that was the deepest visual transect. So if you're a coral reef biologist and you did a transect there to count coral or count fish, that's the same thing that Hudson is doing there, except for the fact that it is at 137 meters depth in Ponape, in Micronesia. When I took that photo, it was the deepest visual transect done by a diver ever. This has been, what, 4 years since we did that. That was a trip to Ponape we did 4 years ago. So after that, we did many transects deeper than that, including one at 150 meters. That's probably our deepest right now, and it's probably going to remain our deepest for a long time, because we don't really, for a number of different reasons, we don't really want to go any deeper than 150 meters in our dives. And it allowed us, so the whole technology, because everything is safer, it allowed us to start doing that. And we started seeing a lot of things on those reefs. The first thing we saw was that those reefs, the deeper reefs, they're not really out of reach. A lot of things that are happening to the shallow reefs, shallow corals in this case, are also happening to the deeper reefs. So because the deeper you go in the ocean, the colder the water gets, there was a general idea, people really suspected, and actually people just assumed that because the water is colder at those deeper reefs, all of the corals at the deeper reefs were safer. However, what we've been seeing is that in some species, there's almost as much bleaching in the deeper reefs as there is in the shallow reefs. And we think that's because, yes, the water's colder, but it's getting warmer. So the same way the water is getting warmer at the shallow reefs is also getting warmer at the deeper reefs. And what really matters for the coral is not the absolute temperature, but it's the temperature variation. So if the temperature, the average temperature goes above what the coral likes, then they bleach. They get stressed out and they bleach. So in the shallows, if they're used to an average temperature of 28 degrees Celsius and it goes to 28.5, 29 for long enough, they bleach. In the deep coral, in the deep reefs here, if they're used to an average of 21 and it goes up to 22, they also bleach. So yes, it's colder, but yes, it's also getting warmer. Something else we saw was that a lot of the other impacts were, for example, this one is a very interesting photo I took in the Philippines. That's about 145 meters depth. And the sea urchin is one of those decorator sea urchin. I don't know what the common name of this thing is, but it just hangs on to everything that's around it. Usually it lives in seagrass areas or in areas with a lot of rubble. And then it puts a lot of rubble around itself to kind of camouflage and look like the bottom. And here, this blue thing attached to it is a piece of plastic, and it's holding on to a fishing line. So we saw, even at those depths, at 145 meters depth, we saw a lot of trash, a lot of fishing lines, a lot of destruction in general. And which showed us that those deeper reefs are really not out of reach. Lots of fishing lines, lots of, in this case here, those lines on the left side of the photo there are anchor lines. So a lot of boats, they know that there's fish on those deeper reefs and they go out and anchor on those deeper reefs. And there is a lot of structure, there's a lot of rocks, there's a lot of corals on those deeper reefs. So the anchors get tangled. And when they pull the anchor, the rope snaps, and then the rest is history, right? It stays in the bottom, destroying the reef for a while as the line goes up and down with the current, and then it stays as trash after that, in this case. So a lot of the same impacts. And at the same time, the transects that we did showed us that even if there were no impacts, those deeper reefs would not be those refuges they were assumed to be, because they're very different. So what we saw was that the community of fishes, when you compared shallow to the several different strata at those deeper reefs, was upwards of 96, 97, 99% different. So it's a community that is almost entirely different than the community we observe in the shallow reefs. So not only we're seeing the same impacts on those deeper reefs, but also the communities of those deeper reefs are very, very different. They're unique in many ways. So unique that we've been collecting all kinds of interesting, cool, new species at those depths. So that's Bart Shepard, another one of our dive buddies in those dives. He's the director of the Steinhardt Aquarium here, and he's collecting a peppermint angelfish there. But you can see that the system itself, this is at about 110 meters depth in French Polynesia. And this system itself allows us to do that. So we could never do this in a submarine unless we spent hours and hours and we used a lot of chemicals, like anesthetic chemicals, to try to collect fish. But it's a nightmare to try to collect a fish with a submarine. But with a rebreather and hand nets, we can collect them relatively easily. So the result of that is that we find new species. Every single expedition we do, everywhere we go, we find new species after new species after new species after new species. So those are just a few examples from the past 3 or 4 years that we've found and discovered and described, or are in the process of describing from throughout the world. Some are from Easter Island, some from the Philippines. There's one from Brazil there that I'm going to talk a little bit more about in a minute. This one is a super interesting one that we described from Zanzibar in Tanzania. And because it was in Africa and because the fish was purple, it reminded us of Wakanda. The first author in the paper, Kai T, a student from the University of Sydney, he came up with the suggestion of naming the fish after Wakanda because it was purple and it had reminded him of the Wakanda color. So we named it after Wakanda, and it got all kinds of media attention because of it. So that's something else we do, is we try to make taxonomy a little bit more exciting to the general public, in order to try to raise awareness. I mean, scientists don't know that those reefs exist, so let alone the public. So the more attention we can bring to those systems, the better. One way we do it is by sound up a little bit more. Even if you're finding my voice annoying and you lowered it down a bit, or maybe put it up again, because I'm going to try to show this video and I'm going to play it now. I'll stop, I'll narrate a little bit, and I'll stop in a few points to tell you what's going on, but try to listen what's going on. You see that one of the results of breathing helium, if you've ever done it in a party, is that like the helium voice, so you hear it in this dive here. Oops, let me go back there. Let's see, how do I play this? There we go. All right, so that's the dive computer. That's on the arm of Mauritius Bell. He's our dive safety officer. He's the one that is doing the video there. This is in St. Paul's Rocks in Brazil. That's at 419 feet depth. So all of these is oxygen sensors. They are reading 1.2. That's the partial pressure of oxygen that we want to be breathing at those depths. It's saying that that number there, stop at 150. It's saying that we have to stop at 150 feet for 1 minute. So that's the first decompression stop. TTS, the bottom right number there, that's time to surface. So we've been underwater for just about 15 minutes at this point. So it takes us about 6, 7, 8, 10 minutes sometimes to get down to those depths. And the TTS, the time to come back to the surface, is already 83 minutes. So that's how long it's going to take for us to come up because of all of the decompression stops. Now I'm going to roll out again. You're going to see, you're going to hear, hopefully, Mauritius screaming on the helium voice there. That's me. I'm trying to collect a fish. Mauritius is saying, look at the shark, look at the shark. And I'm turning to him and I'm saying, no, I want to collect this fish. I'm okay. Wait, I want to collect this fish. So he's screaming, Hudson, Hudson. Hudson's going to turn to him and going to say, I'm okay. I'm okay. I'm collecting this fish. Look up, look up. So I'm going to stop here and I'm going to show you something that probably brings everything to full circle right now. So this is a 6-gill shark. It's a shark that we almost never see in the tropics because it likes cold water. So if you come up here to the San Francisco area, you might see one close to the surface, but in the tropics, in this case, it wasn't St. Paul's Rocks, it's on the equator. You would never see this shark up shallow. They are only deep. They like cold water, so they stay deep with where it's cold. It came up, it looked at us. No other human probably ever saw this shark before. We were the first and probably only ones to see it. But if you look under his pectoral fin there, there's a fishing line. So that long line attached to his mouth there is a fishing line. So that's a fish we've never seen before. A fish that was probably wouldn't go up to the shallows anywhere unless it's cold water. And a fish with already signs of human impact with a fishing line attached to its mouth. And then Mauritius is screaming. Yeah?
We are out of time.
Okay, that was the last one. And Mauritius was screaming to tell us that the shark was coming by. So I want to end by thanking the the committee for inviting me, and hoping we have a nice discussion after. Thank you.
Thank you, Luis, for your nice, very interesting presentation about mesophotic reefs. And now I will ask to Raquel to start her presentation.
Can you see it?
Yeah, it's okay. Yeah, it's okay. It's working.
So I'd like to start thanking you all for the invitation and for being here. I will talk a little bit about customized medicine for corals and some ideas that we have that can, we think, contribute to cope with the coral crisis. I will start as usual with this slide of my dream team, because I always like to present the faces behind all the data. These are the people who are actually producing all the data that I'm going to present. And I'd like also to take the opportunity to thank them all to be such a hardworking, creative, and cool group to work with. And then I go straight to the point. So as we've been discussing during this event, coral reefs are facing unprecedented pressures on a global scale. Increased CO2 emissions are linked with marine heat waves and coral bleaching. And coral bleaching is an example of a dysbiotic process where the symbiotic relationship between the coral and its endosymbiotic algae is broken. And this also affects other symbiotic relationships within these organisms. And although we don't know the exact cellular mechanisms involved with bleaching, we know that increased production of ROS, reactive oxygen species, that precedes bleaching is very likely one of the main triggers of this process. So with that said, and considering that this is basically a dysbiotic process that takes over the coral, and also considering that the algae coral relationship, symbiotic relationship, is not the single relationship that we have in corals and also in other organisms, as also Leticia mentioned, we started to think about ideas of things that could be used or could be manipulated in a way that we could maybe prolongate this health status of corals during a heat wave, or maybe cope with the installed dysbiotic process. In this sense, we started to use some parallels with other systems and other organisms. For example, plant growth promoting bacteria have been used in agriculture systems for many decades. So, and also in humans, and more recently in aquaculture systems, microorganisms have been used to cope with disease, to protect organisms against impacts, or to mitigate contaminants. And so with that, we started to think that maybe also for corals and coral reefs, we should also start to think about a more holistic approach to restore these ecosystems, and to also prevent bleaching or protect them or recover them after a bleaching event. And in this sense, we are looking at something that would be more focused on the host's health rather than the contaminant, rather than the impact. And the premise here is that a healthier organism would cope better with any impact, with pathogens, with increased water temperature, with oil contamination, any different impacts. So we went to the literature and we started to think about mechanisms that could be beneficial in this sense. So the first thing we thought was ROS, because if ROS is the main trigger, we could maybe use ROS scavengers to cope with this ROS, to minimize the amounts, to mitigate the concentration, the excessive concentration of ROS within corals, and maybe that could give us a little bit more time, or could give the algae a little bit more time to stay within corals. In parallel, we could also use other things, because when we have the dysbiotic process started, we have a cascade of several mechanisms and dysbiotic parallel process going on. And so we started to think that maybe we should cope with all these different aspects of the whole process. So we could, for example, use microorganisms that are antagonistic to pathogens, because pathogens are likely to take over and overgrow the native microorganisms and the native microbiome of these corals. So we could use some antagonistic bacteria to cope with these pathogens. We could also feed them with bacteria that could increase the degradation of the MSP that can also be used to attract pathogens. So we could cope with this problem as well, also giving them nutrients and other nutrients such as sulfur, nitrogen, and other things that are important for the algae and other members of the microbiome, and so on. And we came up with an array of mechanisms that could be used in parallel and together, so that we could tackle all these events, these parallel dysbiotic events that take control of corals during the bleaching process. And we came up with this idea, and we proved the concept that that was possible, at least in a lab trial. So what we did is that we selected some of the members of the native microbiome of these corals. So we are not introducing anything new to the reef. We basically isolate these beneficial or neutral microorganisms that are living in association with corals. We bring them to the lab. We select some of these microbes that could be beneficial, that have these different abilities and different mechanisms that together could give us a consortium of beneficial microbes. And then we inoculate them in tanks. And with some of these tanks, we also inoculated with pathogens. Some of them we only increased the temperature, and we evaluated that. What we saw in our first trial is that we could actually mitigate some of the effects of bleaching and pathogens as well. And we had a significant improvement of coral health in this first trial. And this strategy that we are suggesting is, as I said, we bring native bacteria to the lab, we isolate, we do this screening for different mechanisms that can be used together. And this type of approach can be used in different scenarios. So we could use this for bioremediation. So we have a problem already. We have oil impacted coral reef, or we have a bleached reef, and then we could go there and we could give them probiotics and maybe accelerate the recovery period of time, and that could mean that these corals would survive, because time is very important in this case. We could also use this as a prevention, something for some tool of prevention, as we know from NOAA when and where corals are likely to bleach. So we could go there first, we could inoculate these probiotics, and we could expect this reef to react. And in this case, we are doing a parallel with what we do, humans, what we humans do when we need to take antibiotics, and the doctor tells us to take yogurts or probiotics to protect our microbiome. And so we could take it better, the antibiotics. So we would make these corals stronger and make sure that these beneficial microbes are not going to be as impacted as they would be in a normal situation without inoculation of probiotics. And we can also use this in association with other human-assisted evolution approaches. We could use this to increase coral larval settlement. We could increase survival. We could try to increase and make them stronger and healthier, and maybe increase the resilience of these offspring. This is a very short video to illustrate our idea. And so the main concept is that coral reefs without probiotics, when hit by an event of increased seawater temperature, would bleach, at least most of them. Some of them would die. And what we are suggesting is that if we inoculate these reefs with probiotics as a prevention tool, in this case, these microorganisms that would be associated with corals will create this shield and protect them and make them stronger. So a healthier organism is probably more capable of coping with impacts. But how do we apply this to the real world? Of course, and it's very important to highlight that the only solution, in my opinion, the only solution to protect corals and to save corals is, of course, to invest in CO2 reduction and the emission of CO2 reduction. And this is the only solution that I see. But even if you do that, and even knowing that we have to do that, we still have to manage the damage that we have already done. So, and in this sense, we came up with this idea of customized medicine for corals. And why customized? Because it needs to be very clear for all of us that I don't think that coral probiotics can save the reef. I don't think that any of the other palliative solutions that we have can save the reef rather than the reduction of CO2 emissions. But I do think that some of these measurements are necessary for us to cope with what we have now, right? And in this sense, in this regard, I don't think that any of the solutions, that the palliative solutions that we have now are the solution. I do think that we need combined strategies, and I do think that it needs to be very specific for different reefs and different conditions and different situations. And in this sense, I think that just like we do with us, with humans, and just like as we do in bioremediation approaches and any other area in the world, we need to first know more about our reef, our area at least. So this is like a diagnosis. So before we treat a patient, we need to know about this patient, its age, its other conditions associated, other diseases associated with this patient, and diet and everything. So in this sense, we need to know more about the reef we are studying. We need to know also about general mechanisms that are common for all the reefs. So we need to know more about how the microbes are uptaken by corals, how flexible different coral species are, how they interact with the host, how can we manipulate these, how epigenetics also has an important role, and how all these things are connected, and of course, local conditions. So if this reef is also impacted by other anthropogenic actions. So if you have sewage contamination, if you have oil contamination, if you have disease, and if you're looking at, for instance, for the Caribbean, we will select specific solutions that will also take into account SCTLD and everything. So we have the diagnosis of the area, and then we can go for a customized approach. And this is not crazy, because people say, oh, but this is not doable. I think what is not doable is just not doing anything, right? So what is doable and it's effective is that we gotta understand that we need to have general tools that can be used, and we need to have this toolbox full of ideas and full of solutions. And for each area, depending on the specific problems that this area is facing, we will have to combine some of these tools, and we have to apply some of these tools while we work on the most important thing, that is the CO2 emission reduction. So when we have this diagnosis, we can go for this customized medicine that it can include different strategies, or eventually, even if you are only using probiotics, we can have specific probiotics. And I gave the example of the Caribbean. So there we can use probiotics that you protect your organism as a whole, so it makes them stronger, but we can also add some microbes that can specifically cope with SCTLD, as Dr. Valerie Paul and Blake Yuzishima's groups are doing now. So they are developing specific tools, and we can combine the general tools with these specific tools, but we need to have this in mind, that there is no magic pill, there's no magic solution, and that this is not difficult. I think that difficult is to try to keep trying doing things that are not going to work. So we need to have the general tools, and we need to know the areas and apply the most effective tools for that specific area. So we need this comprehensive baseline assessment of the reef health, and we also need the general tools and the toolbox full of tools actually, and then we can go and apply things that can be actually effective. And I will briefly cite the things that we are doing right now because of the short time that I have. I will briefly tackle some of the things that we are doing right now. So we are improving the mechanisms, we are improving the figure and the strategies that we proposed in 2017, because in these last 3 years, we have like a full body of papers and new insights in this field. So we are improving our figure, we are improving our mechanisms and list of mechanisms to suggest other groups, microbial groups, and other mechanisms that can be used. And in this sense, we are also thinking about coral growth, because we do think we can accelerate coral growth with probiotics that are very specifically associated with the calcification process. And these microorganisms can help corals to grow faster, which could be very interesting for coral gardening and other approaches of correlated with restoration. And in this sense, we are looking at the genomes of the culturable fraction that we have. So we are putting together all the isolates that we have in several different labs in the world and looking at the genomes and looking at the beneficial microbes and what they have in common. Also looking at different and new mechanisms that we can explore, and then we can go back to our consortium and make it better, even better. We are looking at multi-domain probiotic consortium. So we are also looking at fungi, and we are also looking at yeast, and combining these different groups that can be used to promote coral growth and health. We are looking at natural traits that we have in reefs. We know that some coral colonies are more resistant, so we tagged them in Brazil, and we've been tracking these resistant colonies, the sensitive colonies. We are looking into the microorganisms associated with them. We know that the microorganisms associated with resistant corals completely different from those that are living side by side from the same coral species and are sensitive. So we are looking at these groups. We are looking at the mechanisms that might be linked with this resistance. We also isolated microbes from these resistant colonies. We developed another coral probiotic consortium for a different coral species, in this case with Muscis media, because our proof of concept was done with Pocillopoda damicornis. And we ran another experiment with these different coral species, a completely different coral probiotic. And again, we had interesting results. So in this case, we had 40% mortality without BMCs, and we had no mortality with BMCs. And we also started to look more deeply into this responses using the metabolomics. We know that we can see significant differences when we look at DMSP contents and lipid contents. And this can also be associated with the tissue, because corals are not dying, corals are not bleaching, and so we have more tissue. It's also linked with the protection and DMSP production or degradation. So we are looking carefully into this new data, but we know that we have more proxies now indicating that the inoculation of BMCs are actually doing promoting preventing bleaching, and also making significant change in this holobiont. We are in this case, and also in other experiments that we are running in the lab, looking at the metatranscriptomics, metagenomics, and the immune response of corals, how corals uptake these microbes, if you can manipulate that as well and make them even more efficient. We are also looking at other basic research to understand better these mechanisms of interaction, because once we manipulate the microbiome, we can look at the responses in a very well-controlled experiment, and we can make sure that the only thing that is different in the different treatments are the microbes that we are giving, and then we can understand more about the mechanisms that are being developed and the mechanisms that are actually triggering this protection. We've been developing a bunch of delivery systems that can be used in the real world. Some of them are inspired in aquaculture and the way they use rotifers and other carriers, live carriers, to feed these microorganisms. So we are feeding corals with rotifers enriched with BMCs, and they actively uptake the rotifers that we drop in the water. And we are running this experiment right now. We are also teaming up with several different people, and I wanted to highlight this collaboration with the Biosphere 2 ocean. It's the largest artificial ocean in the world, and and there we are trying to build a more resilient coral reef and test some of the radical interventions and some of the things that we might need to have in this toolbox in a very near future. We are teaming up with several people from the world. So we created the BMMO network, the Beneficial Microorganisms for Marine Organisms network, where we put together all these amazing people to exchange protocols, to exchange ideas, to accelerate the process of developing this field. And the take-home message here is that the microbiome associated with corals can be manipulated and increase coral resistance and resilience, maybe. We just scratched the surface of this development of this field. There are way more questions than answer. We just started these, and we are having to work and to play in several different positions. If that was something being developed for humans, we'd have people developing and isolating the compound, and other people testing it in small rodents, and some other people with monkeys, until the development of the vaccine in humans. And here we are having to develop everything. That's why we are teaming up with so many amazing people. And the most important thing here is that what I think is that there's no simple solution to solve the crisis faced by coral reefs, and we need to develop and know more about reef health, disease adaptation, resilience, restoration, and have several different strategies and solutions to use, combine efforts and collaboration to apply this in the real work. Of course, we work together. If we work better, if you work together. And with that, I would like to thank all of my collaborators. Some of them are here, and my sponsors. And of course, without any of these people or sponsors, none of these could have been done. Thank you very much.
Thank you, Raquel, for a nice presentation. Would any of you like to ask another panelist or to interact with each other, or we can open to the public questions here? Any questions between you all? No, then I will start with the first questions here. It's addressed to Leandro. Leandro, are you hearing me?
Yes, I am.
Yeah. Edson Vieira is asking, how can we go from cryopreservation of current coral to action on a broad scale for reef restoration in the future?
It's not so simple. As I showed in the presentation, there are so many gaps when we are talking about knowledge on coral gametes, physiology, and biochemistry. However, freezing sperm is not difficult to proceed. Sperm is simple and tiny cell, which normally can be frozen and remain viable after several protocols to adjust the methodology among species. But for the female gamete, the story is completely different. The large size and the amount of yolk in the egg are a barrier when developing a cryo protocol. Apart from that, we will also focus on larvae cryopreservation, which positive results have been reported. I believe reef conservation will only be possible if we work together, as Raquel said, maybe not a particular tool or strategy, but the combination of them. And of course, it's important to society to be aligned with these strategies, being aware of how our habits affect the health of coral reefs.
Thank you, Leandro. Following, we have another question from Rui Kikuchi, Universidade Federal da Bahia, Bahia University. And he's asking, hi, Dr. Godoy, have you tested if the spawned material is viable?
The question, I think that it's addressed to the bleached colonies spawning.
I guess it's after cryopreservation. Is that the spawning material still viable after the crab preservation? I guess.
We still have to perform a lot of tests. As I said, we had just frozen the samples. The previous results we can see so far about 15 to 20% of the cells alive. So the next step, when we can come back to the field during the next spawning season, is to perform these tests.
Okay, thank you. To Letícia, Ana Francisca Tamburos is asking how to control the amount of organisms extracted so that the natural population is not affected. Once collected, do you produce everything else in the lab?
Yes, I think this is a very good point. Actually, to get permits, we have to define how much material we will collect it. And nowadays, the permits are only for like small amounts. But what we do is never, ever like take many, when you have like, we focus on the most abundant species, not the rare ones. So we probably never take the whole colony, just a small portion. And to do this work, this isolation of bacteria and produce everything else in the lab, we generally don't need more than 10 grams of the coral. So it's a tiny, small amount. And you should keep in mind that you should take only the species that are more abundant and never take the whole colony, but just a small portion.
Thank you. We have another question. I will split to Luis Rocha now, then I will back to Letícia too. Luis, are you hearing me, please?
Yeah.
Yeah. Phil Carp is asking for you. With a rebreather, is there still an issue of nitrogen buildup and the associated need for the compression stop?
Yeah, good question. So the reason why we need to decompress when we go very deep or when we stay long underwater is because the gas that we're breathing gets dissolved into the tissue. And if it comes out of the tissue very quickly, it becomes bubbles. And then if it becomes bubbles, it stops the blood flow, and it's really bad. I mean, it can stop the blood flow to our heart, can stop the blood flow for our brain. The problem we solve with nitrogen is the other physiological interaction. So nitrogen, when we breathe nitrogen under pressure, in addition for it to being in the tissue and then coming out as bubbles, it also has physiological interactions that make us feel drunk, basically, as if we're drinking alcohol. So we replace the nitrogen with the helium so that we don't feel drunk. But the helium has the same problem of decompression as nitrogen. So helium is still a gas, and it goes into the tissue and it still has to come out of the tissue. So when we're doing decompression, when we go very deep, we remove the nitrogen and we add helium, but we still have to decompress for the helium. So the decompression is about the same. It doesn't change. What does change is that when we are at depth and we're breathing mostly helium, we're not feeling drunk, which is a lot safer. I can tell from experience.
Thank you. Now we are going to Raquel. Raquel, Alessandra Paul is asking to Dr. Peixoto, is there evidence that the same isolated microbes could be both beneficial or pathogenic depending on other environmental conditions? Like commensal bacteria of the human microbiome can cause good dysbiosis under certain conditions.
In our case, for our selection, we exclude any potential pathogens. So anything that could be commensal and pathogenic, like some vibrios and some other genera or species that can be or are known to be even potentially pathogenic, we exclude from our screening. So we are only using things that are known for being very abundant or very widely distributed, and that have only reports as being beneficial. So I'm not aware of anything among these groups that could be potentially pathogenic. What we do is that we always test it in the lab. So we increase the temperature, we change the conditions, and we track it. And once we see that it's safe in our experiments, then we think that this is, we elect this as a potential beneficial consortium. So, and we also look at the genomes and anything that could be potentially harmful. But no, I'm not aware of anything that is pre-selected that could be pathogenic.
Thank you. Letícia, Alessandra Polo is asking, assuming corals need to fight pathogenic bacterial infections, is there research being conducted on possible antimicrobial compounds derived from corals?
Yes, we do in our lab. We have a collaboration with Dr. Gabriel Padilla lab and we are searching for antimicrobial compounds that can be used against multi-resistant bacteria like MRSA, that is methicillin-resistant Staphylococcus aureus, and vancomycin-resistant Enterococcus, and several others. But Gabriel also is doing some research trying to test one bacteria against the other, both isolated from the corals, to see whether one bacteria can be good as a antibiotic producers, looking from a coral perspective. So we try to do from the human health perspective and also from the coral health perspective. Yes, we do.
Yeah, thank you. Another question to Dr. Godoy from Melina Ferreira, a Brazilian from Encontro Recifal Brasileiro. She's asking, are gametes from populations living in warmer places for cryopreservation being selected, assuming they are locally adapted and more resistant to high temperatures?
Not yet. Our project is quite recent. We have been working for 1.5 years, so a lot of work has to be done. In this first stage, we are exploring the basic aspects of the gametes biology and physiology, assessing their sensitivity to lower temperature to achieve a successful cryopreservation protocol. So far, our studies have been conducted at Recife de Fora Marine Park, since the research base of Coral Reef is located there. But yes, we can use these approaches when the cryo protocol are established.
Okay, thank you. Now, Raquel, some questions here. I will choose someone. Selwall, it's a nickname, asking if it's possible that some coral species benefit more than others in the same reef, and that could cause some changes in the community dynamics. You're using your protocol. Your audio is off.
We've been trying to select some, now we are studying, so I will also take the opportunity to answer another question that I saw here. So at this point, our consortia are all species specific, but we are now working in a project where we are trying to select some groups that could be beneficial for most of the coral species, at least from some areas. So the idea is to go and try to select and come up with 1 or 2 or maximum 3 cocktails that could reach all the potential or the most important reef builders in an area. And in this sense, it's, of course, it's considered a radical solution, and it's only going to be used whether we have a radical scenario, so a very damaged reef. So in this case, if we could save at least some of the species, I think it's better than not saving any of them. But what we are trying to do is that we are trying to tackle as many as we can. And we are also working in a different project where we are also looking at other members of the reef. So we are looking at sponges, fishes, and all other organisms that can be affected by this bacteria. But again, We are using bacteria that are very commonly found in the reef. They are native from that reef, and they are already there. What happens is that when you have an impact, they are affected as well. And so one of the things that we think is that we are trying to reintroduce these bacteria back, or at least keep some of them in a condition that not pathogens are not going to overgrow these beneficial or eventually neutral microbes.
Thank you. Luis, we have a lot of questions about here. I will try to put all of them together, but people are very curious about the cost of the equipment. I will try to synthesize here, but it's about, let me see where it is. It's here. People are asking, I will put 3 or 4 questions very similar to other. Rocha, do you think that here in Brazil, our students are prepared, trained for scientific dives, in this case, for deep dives? And how do you think that should be to return of scientific dives after the actual pandemic of COVID? Asked by Sarah Butrosi Kennedy. And Thais Macedo is asking if there are women doing scientific diving in deep reefs using rebreathing.
Okay, I'll answer the quick one first. If there's any women doing it, to the same depths that I go, there's one woman, Sonia Rowley from Hawaii. She's the only one doing this. I mean, everybody knows that science in general is more male than female. Diving in general is more male than female, and technical diving is almost overwhelmingly more male than female. There's very few women doing it at the recreational level, at the scientific level. It's very sad, because it's a different perspective that we're missing. And I don't exactly know what the reason is, but I think in the case of deep diving, I think the reason is because there's very, very few opportunities to do it. We have one postdoc, not in my lab, in Pim Bungertz. Pim Bungertz is one of our colleagues here at the California Academy of Sciences that works on mesophotic coral ecosystems. He's working on corals, not fish. And he just started his diving program. He has one postdoc that is diving with him to about 60 meters depth. So the certification has 3 different categories. The first one, you just use the rebreather. The second one, you can use the rebreather to about 60 meters depth, which is what Pim and his female postdoc is doing. And then the next one is all the way to wherever you want to dive in, which is the one I do. But Pim is more interested in the 60-meter depth. So his postdoc, Alejandra, she's doing it at that depth. There's a few others doing it at depth, but this depth that I go to, 150 meters or so, there's only one that I know of, Sonia Rowley. I wish there were more. If you want to come work in my lab, let me know. Part of the problem I have in my lab in particular is because it takes a long time, a very long time. It can take up to 2 years to train someone just for the diving, not even for the science, to do this type of diving. And there's some guys that already come with the training that they did because they like diving or something like that. And I haven't found a woman that has that yet. And I hadn't found a woman that even wants to do it during the dive. We're training all of our female, we have a bunch of female dive officers here at the Academy. We're training them. And they are competent to dive at those depths, but just from the dive safety side, not from the scientific side. The other question was if this can be done in Brazil safely. It can be done safely, but it's very hard. It's very hard to do it here safely. It's 10 times harder to do it safely in Brazil because of the expense, because the rebreathers are big things that have to be serviced every year. And the company that services them for us is nearby here, so we can just drive the rebreathers there, drop them off there, and have them serviced and sent back to us. If you're in Brazil, you have to send them here for service. and it takes months and months to go through customs and everything. It's just a nightmare. But even if you go through all of that, there's still the question of scientific diving in general, which is a very tightly controlled activity here in the US, and in Brazil it's not. And whenever it's not tightly controlled like that, accidents happen. So it's very difficult to do it in Brazil. There was a second part for that question. I can't remember which it was anymore.
People asking about the cost.
Go ahead. The cost.
Costs.
Okay. It's expensive, but it's cheaper if I was doing it just using normal scuba diving with helium, because of the cost of helium. So one of those rebreathers cost between $7,000 and $10,000, depending on what manufacturer it is. But I would spend that in helium, just in helium, in one expedition, if I was using it, just breathing and exhaling it as bubbles. By using the rebreather, we use a lot less helium, so we save on the helium, and then we invest that upfront in the equipment, and then the equipment lasts for several different trips. So in the long term, it's savings. So yeah, it's a high initial investment, but it's worth in the long term.
And how have the advanced technical dive influenced in the scientific production, mainly of those organisms from mesophotic zones?
Right,
right. Is there an increase of works in these habitats because of this technique?
Absolutely, yes. There has been an explosion almost, I think, in the amount of papers published by about mesophotic ecosystems, ecology, corals, fish, algae. There's a lot of people studying them throughout the world now because of the popularity of rebreathers and them becoming safer and safer over the years. The rebreather technology itself is very old. It's even older. It predates normal scuba gear, but it's never been as safe as it is now, with all of the new computers and the new sensors and the computer that goes in your face and all of that. Those weren't here before. So now with all of that, it's becoming more and more popular. There's a website called mesophotic.org. And if you go there, it kind of summarizes all of the literature and shows you everything that was published, where, and how it's been increasing exponentially over the last few years or so. This term mesophotic was first proposed about 10 years ago. Before that, people used to just call it deep reef or twilight zone or something like that. But since it's been proposed, I'd say at least maybe 500 papers published about it.
Yeah, thank you.
We are running out of time for this session, but I think we were able to answer a lot of questions you guys had. It was really, really interesting. We have another session starting in about 15 minutes. And I'll tell you a little bit about this. Here we go. Get to the next. There we go. So we have a session from 3 till 4 on management and conservation, looking at the impacts of coral reef management and the impact of sargassum. And tomorrow we have another plenary talk from shallow reefs to deep sea. So if you like Dr. Rojas' talk today, you will love Diva Amon's talk tomorrow, where we go even deeper down into the depths of the Caribbean Sea. And there's also going to be some programming tomorrow on the Brazilian side. And I'll let Cesar tell you a little bit about that.
Hello, everyone. I'll just send you a quick remember about our plenaries tomorrow. They'll be held in Portuguese, unfortunately, but we can later on try to use the captions from Google and from YouTube and try to catch up with the presentation. So everybody's invited and you can join us tomorrow. Thank you. And thank you all the speakers today. Thank you, Francisca. Thank you, Chelsea.
Thank you.
Speakers from us as well. All right. So I'll see you. if you want to come to the next talk. Thank you, everybody.
Thank you.
Thank you.
Thank you.